So hello everyone, we're Seafeed. My name is Amanda and my group members are Ryan, Lourdes, and Denise. So fresh water is crucial for human beings, but it's currently an overburdened and rapidly depleting resource. On top of that, there are many factors that are reducing the amount we have available, such as global population growth, saltwater intrusion into our aquifers, and high depletion rates. To mitigate this, we've developed an indoor macroalgae cultivation system for growing and harvesting seaweed. You can see a picture of our prototype to the right. We chose to grow seaweed for various reasons. Macroalgae that's controlled and sustainably cultivated is looked at as the future method for supplying biomass to meet market development needs. It can grow in fresh, salt, and wastewater and can serve as a biofuel, as a bioactive component, as food, or as animal feed. By expanding its production, growing seaweed can lead to a reduction in land and freshwater use. Additionally, based on our talks with researchers like the Aquaculture Innovation Center, we've also determined that there's a gap in the existing research market for macroalgae cultivation. These are all spaces in which we feel we can make a positive impact.
For our scientific component, we had to understand how to connect the tanks and achieve consistent water flow using fluid mechanics principles. We wanted to arrange the pipes in a way we could have the same water flowing throughout the structure and have a consistent water height. The pipes are designed to take water up from the reservoir with a pump and into the top tank. Then the water falls into the second tank and flows back into the reservoir. The top two tanks are our growing tanks, and the reservoir on the bottom maintains the water quality. To test for optimal growing conditions, we chose our variable to be lighting. We control the LED lights of each tank separately and manage. Hello, can you guys still hear me?
Yes.
Sorry, my Zoom call failed out. Okay. One of our main contributions is an indoor farming system. An indoor system means we can control temperature and produce growth all year round, no matter the location. In order to succeed, we had to understand how to properly maintain the water quality, as two nutrients, temperature, pH, oxygen flow, and lighting are significant roles in the seaweed life cycle. Our seaweed of choice was Ova lactuca because it is easy to grow, free-floating, easily accessible, and to prove our indoor concept will work with other free-floating species. For our technology component, we wanted to monitor our pH and temperature regularly. In order to do this, we needed a Raspberry Pi to be constantly running, gathering data. On the right, you can see our technology subsystem. Our sensors are connected to the Arduino board, which is connected to the Raspberry Pi. In order to export the data, we created a Python file that would read the data from the Arduino serial monitor and export it into a continuously updating CSV file. Our other technological component is our variable. The LED strips were attached to the lids of our tank and plugged into timers that have a 24-hour clock in which we can decide the hours we want it to be on and off.
You can see in this video how all of the factors come together. The structure is 8 feet tall and currently operating from my home garage. The reservoir contains all of the sensors used to manage the water quality. The technology hub located in the back is connected to our pH and temperature sensors, which collect 5 readings every 10 minutes. These values are then averaged and displayed in a continuously updating CSV file. In this way, the user can see if the water quality is out of range and manually adjust the factors using our system's temperature heater.
We identified 3 systems that had similarities to our project. The picture on the left shows Monterey Bay Seaweed site in California. The center picture shows the IEEE growing system, and the right shows the tubular system from the Rutgers Aquaculture Innovation Center in Cape May, New Jersey. So here's how we're different from the competitors. Monterey Bay Seaweeds is an outdoor macroalgae growing system that primarily uses its seaweed for restaurants. It also has a direct connection to the ocean. IEEE has an outdoor macroalgae system, and it has nutrients, pH, and temperature being monitored by Arduino. The AIC is an indoor microalgae growing system that has a horizontal scalable tubing network, and its nutrients and pH are monitored. How we're different is we're a first of its kind macroalgae growing structure, and our tank has a system that's temperature controlled and closed loop. So a little bit about our innovation. We have a lighting system that utilizes timers that allows us to have settings to mimic natural light conditions. We are monitoring some of our parameters like lighting and pH to see which factors most affect our experiment. We've collaborated with experts such as Dr. Bhattacharya, who's considered an expert in ova-lactuca. And finally, we've created an indoor design and made it scalable structure to make it versatile and marketable.
So our successful project looks like a functional structure that grows seaweed to generate insight. A functional structure meaning that our infrastructure is sufficient to support seaweed growth, growing seaweed meaning that our water quality is adequate for ova-lactuca, and generating insight meaning that we have a method of collecting data, and the method translating data to analysis is generalizable for any type of macroalgae. For validating and testing, we have validated our structure is functional, and we began our first experiment as of yesterday for growing seaweed. In terms of generating insight, we will sanity check the data and seek external feedback from Aquaculture Innovation Center members. For project management, throughout these 2 semesters, we've been sharing responsibility to ensure that all projects are completed, and we've been holding each other accountable through weekly group meetings and through frequently meeting with Dr. Defari. In terms of risks, about 2 weeks ago, several group members contracted COVID-19, which understandably delayed our project by about a week and a half. Thankfully, we were able to speak with Dr. Defari, and he was very accommodating with extending our deadline. In terms of other risks, our structure had to be constructed virtually in the lab, and the lab technician was available 6 hours per week, which was a time constraint. In addition, we had to transport the structure from the lab to our group member's house. We had to source the seaweed, which was not straightforward, and we are currently running experiments to ensure we have this experiment count. In conclusion, freshwater depletion is a major problem in today's world, and macroalgae can help supplement existing freshwater using resources in the agriculture and energy sectors. There is currently a research gap on how to grow macroalgae indoors, and where our project fits in is that it is a proof of concept that macroalgae can be grown indoors, and a jumping off point for interested parties like academics and industry members to develop their own systems and explore this sphere.